Imagine this: you're running a marathon, your legs are burning, your lungs are heaving, and you're desperate for the finish line. Suddenly, your body's energy production grinds to a halt. Total shutdown. Your muscles can contract, your brain can send signals, and even the beat of your heart falters. This is what happens when cellular respiration fails. Hello, erudites. It's me, Kim. For this session, we will be discussing the long process of cellular respiration, particularly its purpose, steps, and how it plays a major role in many aerobic and anaerobic organisms. This is actually a really fun and important topic once you understand the whole thing. So, without further ado, let's dive right in. So, for our presentation outline, we will be first defining what is cellular respiration, the two types of cellular respiration, the inputs and outputs, the three major stages, the total number of ATP produced, the anaerobic respiration and fermentation, and the two types of fermentation, as well as the brief summary. So, what is cellular respiration? So, based from the definition, it is a metabolic process that converts biochemical energy from nutrients into ATP or adenosine triphosphate. ATP is basically the energy of the cell. Its purpose is to generate, as I've said, usable energy for all life processes, including cell movement, cell growth, cell repair, and cell transport. The overall chemical equation for cellular respiration includes glucose and oxygen, which are then converted into carbon dioxide, water, and energy in the form of ATP. So I have provided a diagram here of the entirety of the cellular respiration. So the food that we intake and the oxygen that we breathe, this basically goes inside the cell. Now, what the cell is going to do is to basically convert the glucose from food into ATP, as well as produce the products which are carbon dioxide and water. So, in order to do this, we're going to go through three main stages. First is the glycolysis, second is the Krebs cycle, and third, which is the electron transport chain. Before we proceed with the three stages, it is also very important to discuss the two types of cellular respiration. First type is the aerobic respiration, and the second type is the anaerobic respiration. So, for aerobic respiration, this basically requires oxygen. From the word aero itself, this means air. So, air is involved. Next, it occurs in the cytoplasm and the mitochondria. It also yields a very, very large amount of ATP, approximately 30 to 32. But this sometimes depends on how much or how many electrons are produced in the third stage. Next, we have the anaerobic respiration. So from the uh, word anaero, this basically does not require oxygen. So no oxygen is involved in this process. It occurs also only in the cytoplasm and it yields very little ATP, only two, producing byproducts like lactic acid and ethanol, which we will discuss later on. Next, for the input and output of cellular respiration in aerobic organisms. First, we have the primary inputs or the reactants. We have glucose from the food that we take, as well as the oxygen from the air that we breathe. So, from glucose and oxygen, this basically produces our primary outputs or products, which are the carbon dioxide, the air that we exhale, as well as the water and ATP. It is also important to mention on the energy carriers or electron carriers NADH and FADH2 because they are crucial intermediate products that carry high energy electrons to the final stage. And it is also very important to take note of these two electron carriers as we go along the discussion. Okay, so for the very first stage, we have the glycolysis. From the word lysis, meaning breaking down, and from the word glyco, meaning sugar, it is basically the breaking down of sugar or the breaking down of glucose in particular. It also occurs in the cytosol, the jelly-like fluid of the cell. For its key features, glycolysis is anaerobic. It can occur with or without oxygen, making it a universal and ancient process of life. Its main goal is to split a glucose molecule into two smaller molecules called pyruvate and generating a small amount of ATP and crucial electron carriers. For the first phase of the glycolysis, we have the energy investment phase. Before we can harvest energy, we have to spend a little. Think of this as the priming the pump stage. For our input, we start with one molecule of glucose, a 6 carbon sugar, and two ATP molecules. Remember, ATP is basically the energy. So for the process, the cell uses those two ATP to add phosphate groups to the glucose molecule. This addition of energy makes the glucose very unstable and activates it for the splitting that comes next. For the output, the result is one molecule of fructose 1,6 biphosphate, which is still 6 carbons, but now it's energized with two phosphate groups and ready to be cleaved in the next phase. Next, we have the cleavage phase or the second phase, the splitting. The main event here is splitting the 6 carbon molecule fructose 1,6 biphosphate. It's then split into two identical 3 carbon molecules known as G3P or the glyceraldehyde 3 phosphate. The output of this phase is two molecules of G3P which are now ready to generate energy. Now for the third and final stage, which is the energy payoff phase or the harvesting stage. Since we now have two G3P molecules, all reactions in this payoff phase happen twice. Two critical things occur. First is the redox reaction. Electrons are removed from the G3P and loaded onto the carrier NAD+, producing NADH. This NADH is the valuable electron carrier that will be used later for massive energy production. Next is the ATP production. Phosphate groups are removed from the G3P derivatives and directly transferred to ADP to form ATP. We call this substrate level phosphorylation. Now you might be wondering, how does ADP turn into ATP? So basically, ADP means adenosine diphosphate. Di meaning two. It only has two phosphate groups. Now, since in our glucose, we've attached phosphate groups a while ago. So now those ADP, they will get those inorganic phosphates, and then since they will get those, it will add another phosphate to ADP, which makes the phosphate groups now 3, making the adenosine triphosphate. After those three phases of glycolysis are complete, the cell has produced quite a few things. 1. The two molecules of pyruvate. These are 3 carbon molecules that will move on to the next stage of cellular respiration. Next, we have the 4 molecules of ATP. This is the total ATP generated from the payoff phase. And last, we have 2 molecules of NADH, or critical high energy electron carriers. Since we had to spend 2 ATP in the initial investment phase, the overall profit or the net yield of glycolysis is 2 ATP, 2 NADH, and 2 pyruvate. This entire process can also be summarized by the simple equation glucose plus 2 ATP plus 2 NAD plus 2, 2 ATP plus 2 NADH and 2 pyruvate. Now, to explain through visuals, I have here provided a diagram for the whole entirety of the glycolysis. So, for the cyan dots, this basically represents carbon, and the orange dots, this represents the phosphate. So, una muna, we have the glucose, right? So, the glucose, it's a 6 carbon molecule. Ngayon, it will undergo phosphorylation stage, which basically means you're going to donate phosphate groups. Now, how do you donate? Basically, if you're gonna donate phosphate groups, you're gonna make use of energy, which is in the form of ATP. So ngayon, itong phosphorylation stage na to, you basically need 2 ATP. So now, if you're gonna make use of 2 ATP, you're gonna donate 2 phosphate groups. Ngayon, once you've donate the 2 phosphate groups, you'll be you're going to create a new molecule called fructose 1,6 biphosphate. And this molecule is actually very unstable because of the energy na nanggagaling sa ginamit mo na ATP. Ngayon, it will undergo lysis, which means splitting. Hatiin siya sa dalawa. So, ang mangyayari, ang maging result is itong triose phosphate. Ngayon, ito yung triose phosphate. Before oxidation, kukuha siya ng isang inorganic phosphate. So, as you can see, from 1 phosphate, magiging 2 phosphate na siya. So, ang mangyayari, sa isang triose phosphate, dalawang phosphate na siya. Ngayon, after that, kapag dalawa na yung phosphate niya, it will undergo oxidation. So, how does oxidation happens? Ngayon, itong NAD plus na to, these are basically electron carriers. Ngayon, since mag-oxidize siya, yung NAD plus, kukuha siya ng electrons and hydrogen sa may carbons. Tapos, once nakuha niya yon, ma-oxidize and then from NAD plus, magiging NADH na siya. So, this also happens sa isa pang triose phosphate. So, basically, ang mangyayari dito is um, makukuha, makakakuha tayo ng dalawang NADH. After that, after ng oxidation, we have the ano na, G3P, which is yung glycerate 3 phosphate. Ngayon, it will undergo the phosphorylation. So, ito yung opposite na process ng phosphorylation. 'Di ba sa phosphorylation, we're basically adding phosphate groups. Ngayon, we're gonna remove them na. Kasi ito yung phase na energy harvestment. Ngayon, sa dephosphorylation, itong 2 ADP na to, itong ADP na to, kukuha siya ng isang phosphate group. Ngayon, since we have 4 phosphate groups, 4 ADP is ready to get those. Ngayon, ang mangyayari, ang maging result dito sa phase na to is 4 ATP will be made. Pero, you might be confused since ang net yield natin is 2 ATP. Remember yung 2 ATP na ginamit natin nung una. Basically, ang mangyayari is 4 minus 2, 2 ATP na lang talaga yung nakuha natin as well as 2 NADH. For the second stage of cellular respiration, we have the Krebs cycle, also known as the citric acid cycle. So, what is this Krebs cycle all about? It's basically the central metabolic pathway that completes the breakdown of the remnants of glucose. Where does it happen? This happens inside the matrix of the mitochondria, the innermost compartment of the powerhouse of the cell. Unlike glycolysis, this cycle is strictly aerobic, meaning it requires oxygen. Why? Because it relies on the electron transport chain, aka the final step, to regenerate the NAD plus and FAD molecules it needs to keep spinning. So its main goal is to completely oxidize the pyruvate derivatives, stripping them of all their remaining energy. This releases carbon dioxide and generates a modest amount of ATP, but more importantly, it produces a large number of electron carriers, NADH and FADH2, which will power the cell's main energy production later on. Before we jump into the Krebs cycle, we must cover the quick but critical transition step, the pyruvate oxidation. This process bridges glycolysis and the cycle itself. So, basically, in pyruvate oxidation, it starts with the two molecules of pyruvate that came out of glycolysis. This happens as the pyruvate moves from the cytosol into the mitochondrial matrix. For this process, each pyruvate molecule is converted into an acetyl-CoA molecule in two main actions. First is the decarboxylation. One carbon atom is removed from pyruvate and released as carbon dioxide. This is where we start breathing out the carbon from glucose. Second is the redox. Electrons are captured by NAD+, forming another variable carrier called NADH. Now, since we start with two pyruvates, the total output for this step is 2-acetyl-CoA, 2 carbon dioxide, and 2 NADH. The acetyl-CoA is now ready to feed into the Krebs cycle. Next, for the cycle itself, this is where we fully break down the fuel. Remember, the cycle turns twice for every one molecule of glucose. For our input, the cycle starts when 1-acetyl-CoA, the 2-carbon fuel, the product of the oxidative phosphorylation, combines with a 4-carbon molecule called oxaloacetate. This forms the 6-carbon starter molecule called citric acid. For the process, the citric acid then goes through a series of reactions. The main job is to completely oxidize the carbon atoms, stripping their energy and releasing them as waste. First, we have two molecules of carbon dioxide. Carbon dioxide are released. This eliminates the last of the carbon from the original glucose molecule. Next, we have the carrier production. This is the most crucial step. We generate a massive number of electron carriers, 3 NADH and 1 FADH2. These carry the high energy electrons to the final stage. Lastly, we have the ATP production. One molecule of ATP is generated directly via substrate phosphorylation. The output. Basically, the cycle finishes by regenerating the oxaloacetate so it can accept the next acetyl-CoA and keep the process going. The final output per acetyl-CoA turn is 2 carbon dioxide, 3 NADH, 1 FADH2, and 1 ATP. Now, for the net yield of Krebs cycle, we have 4 carbon dioxide from the cycle itself plus 2 carbon dioxide from the pyruvate oxidation. So in total, we have 6 carbon dioxide. Next, we have the 6 NADH from the cycle itself plus 2 NADH from pyruvate oxidation, which means we produced 8 NADH in total. Next, we have created 2 FADH2 as well as 2 ATP. Here, I have provided a diagram of the Krebs cycle. So, from glucose and fatty acids, we'll be producing the acetyl-CoA. Now, this acetyl-CoA, we will produce a product called citrate. So, in this step, the pyruvic acid from glycolysis is broken down into a 2-carbon molecule, releasing one molecule of carbon dioxide. During this process, high energy electrons are transferred to NAD plus to form NADH. Next is the isocitrate. This enzyme catalyzes the isomerization reaction by removing and then adding back the water to yield isocitric acid. So our product is now called isocitrate. Next, we have the acetoglutarate. In the first oxidation step of the Krebs cycle, isocitric acid is oxidatively decarboxylated to a ketoglutaric acid. Next is the succinyl-CoA. This is a complex of different enzymatic activities similar to the pyruvate dehydrogenase complex. Our product here is succinyl-CoA plus carbon dioxide plus NADH. Next is the succinate. Succinyl-CoA, like acetyl-CoA, has a thioester bond with very negative free energy of hydrosis. The reaction is GTP plus ADP to GDP plus ATP. Our product here is succinate plus GTP, or also considered as the ATP. Next is the fumarate, oxidation of succinate to fumarate. This is the only citric acid cycle that is tightly bound to the inner mitochondrial membrane. It is an FAD-dependent enzyme. Next is malate. The fumaric acid reacts with molecule of water to form malic acid in the presence of the enzyme fumarase. And lastly, the oxaloacetate. The oxidation of malate to acetate. It is an NAD plus dependent enzyme. So in this process, we're going to be able to produce 3 NADH, 1 FADH2, and 1 ATP, only in just one single cycle. But the Krebs cycle is actually composed of two cycles, since we have two acetyl CoA. Now, for the final and last stage of the cellular respiration, we have the oxidative phosphorylation. So, what is this? This is the body's primary mechanism for producing ATP, or cellular energy, utilizing the energy stored in NADH and FADH2, which are electron carriers. So, where does this stage happen? This happens in a specific location in the mitochondria, specifically in the inner mitochondrial membrane. For its oxygen requirement, it absolutely requires oxygen, making it an aerobic process. This is because oxygen serves as the final electron acceptor. The main goal of the stage is to take the energy from NADH and FADH2 to establish a high concentration of protons, or an hydrogen gradient. This gradient then powers an enzyme to synthesize the largest amount of ATP we see in cellular respiration. Now, for the first part of two parts of the oxidative phosphorylation, we have the electron transport chain. The inputs are the electron carriers NADH and FADH2 derived from glycolysis and the Krebs cycle, and crucially, oxygen from breathing. The process starts when NADH and FADH2 drop off their electrons at protein complexes embedded in the inner mitochondrial membrane. As these electrons move down the chain, like falling down a staircase, they release energy. This energy is immediately used to pump protons, hydrogen ions, out of the mitochondrial matrix, building a high concentration of protons, or a proton gradient, in the intermembrane space. The output is this proton gradient, which will be used to make ATP. The spent electrons, having completed their journey, are then handed over to the final acceptor, oxygen, which combines with protons to safely form the byproduct, water. As you can recall, water is also one of the byproducts of the cellular respiration. Now, for the second part of the oxidative phosphorylation, we have the chemiosmosis. The inputs of this part are the proton gradient we just created, along with ADP and inorganic phosphate. The process uses the strong electrochemical gradient. Protons flow back down their concentration gradient through a specialized enzyme embedded in the membrane called ATP synthase. As protons pass through, this enzyme physically spins, taking ADP and adding a phosphate group to it. The output is the generation of a large amount of ATP. Overall, the net yield of oxidative phosphorylation per glucose molecule is approximately 26 to 28 ATP, with water as a harmless byproduct. This makes it by far the most efficient ATP producing stage of the entirety of the cellular respiration. Here I have provided the diagram of the electron transport chain as well as the chemiosmosis. So we're going to be explaining this diagram. So we know that yung electron carriers natin, which is yung NADH and FADH2, these are the products of the glycolysis and the Krebs cycle. Ngayon, they are electron carriers. Since they carry electron, i-deliver nila yung electron sa may protein complexes na to, yung number 1, 2, 3, 4. After that, after na ma-deliver, bale, para siyang pathway, yung electrons, para siyang magta-transport sa mga uh, protein complexes na to, and the energy of this transportation is then used to pump the hydrogen protons or the hydrogen ions sa may cellular mitochondrial matrix. Ipapump nila yung hydrogen doon, hydrogen ions, papunta sa may intermembrane space. Ngayon, once this hydrogen ions gets pumped sa intermembrane space, dadami sila, tataas yung concentration kasi maraming hydrogen ions. Ngayon, since sobrang taas ng concentration ng gradient na yon, syempre, through the process of diffusion from high concentration to low concentration, gusto nila bumaba pabalik sa may rather sa may cellular matrix. Ngayon, paano sila makakabalik doon? So, we actually have this very enzyme complex called ATP synthase. Bale, ito yung dito sila travel pabalik sa may cellular matrix. Ngayon, kapag papasok yung hydrogen ions na to sa ATP synthase, this basically turns the ATP synthase, and then yung rotation na to, it helps catalyzes the ADP and an inorganic phosphate to produce ATP. Kaya marami tayo na-produce na ATP sa stage na to, which is yung oxidative phosphorylation. Now, to finish our review of aerobic respiration, let's look at the grand ATP tally per single glucose molecule. First, glycolysis contributes a net of 2 ATP. The Krebs cycle adds another 2 ATP, and the powerhouse, the oxidative phosphorylation, which includes the electron transport chain or ETC and chemiosmosis, yields the vast majority, providing approximately 26 to 28 ATP. This brings the total or the grand total to roughly 30 to 32 ATP produced from every glucose molecule, confirming aerobic respiration as a highly efficient energy process. Next, we'll be tackling on the anaerobic respiration and fermentation. These two processes that happens in anaerobic organisms. As you know, we've discussed about the cellular respiration that happens in aerobic organisms. Now, we'll be discussing on what happens in anaerobic organisms. Without further ado, let's start. So for anaerobic respiration, this is the process that still involves the main stages of cellular respiration. We have glycolysis, Krebs cycle, and an electron transport chain. The key difference from aerobic respiration is in that final step. Instead of oxygen, anaerobic respiration uses an inorganic molecule like sulfate, nitrate, or sulfur as the final electron acceptor in the ETC. As you know that yung kanina, yung cellular respiration sa may ETC, ang oxygen is yung final electron acceptor. Moving on, let's look at fermentation, another anaerobic or no oxygen metabolic process. Like anaerobic respiration, it starts with glycolysis. However, it stops there, not using the Krebs cycle or an electron transport chain. This results in a very long energy yield, a net of only 2 ATP per glucose molecule. The main job of the steps after glycolysis is crucial to regenerate NAD plus from NADH. This allows glycolysis to keep running and produce that limited ATP. Finally, in fermentation, the final electron acceptor is an organic molecule like pyruvate. This process gives us many common products. For example, lactic acid fermentation by bacteria, and while alcoholic fermentation by giving us products like bread, wine, and beer. We'll be diving in those two types of fermentation after this. Okay, so for the two types of fermentation, we'll start first with the first one, the lactic acid fermentation. So what happens in this process? In this process, the pyruvate molecule that's produced from glycolysis is converted directly into lactate, which is a form of lactic acid. This process is critical in two main areas. Number one, it occurs in our human muscle cells when we're performing very strenuous activity and can't supply enough oxygen. Second, it's also how certain bacteria and fungi work, specifically the ones we use to create staple food products like yogurt and cheese. Next, we have the second type of fermentation, which is the alcohol fermentation. In this anaerobic process, the pyruvate produced from glycolysis is converted into ethanol, which is a type of alcohol, and carbon dioxide is released. This process is very critical to humans because it's carried out by yeast and some plant cells. It's the mechanism we use for making bread, baking bread, where the carbon dioxide makes the dough rise, and for brewing beer and making wine, where the ethanol is the desired product. Now, to summarize our discussion, remember this: cellular respiration is a controlled multi stage process that efficiently converts stored energy into usable ATP. We covered two main strategies for energy. Aerobic respiration maximizes the ATP yield, producing 30 to 32 ATP using oxygen, primarily inside the mitochondria. Second, anaerobic respiration, like fermentation, provides a rapid but low yield source of only 2 ATP in the absence of oxygen, operating solely in the cytoplasm. The two main types of fermentation are lactic acid fermentation, which produces lactate, as seen in our muscle cells, and alcohol fermentation, which produces ethanol and carbon dioxide, carried out by yeast. Ultimately, the vast majority of all cellular energy is produced in the mitochondria, solidifying its role as the central powerhouse of the cell. And that ends our discussion. This topic is very important because it explains the fundamental process by which all living organisms convert the chemical energy found in food, like glucose, into usable cellular energy known as ATP or the adenosine triphosphate. Without this process, cells wouldn't have the energy to power vital functions like movement, growth, and repair, which would ultimately lead to the death of the organism. With that, thank you for listening and good luck, erudites.